Hello, and thank you to everyone around the world for joining us for this week's lab meeting. My name is Kristen Abood. I am the science editor at the Human Vaccines Project, and I will be your moderator today. Although the BA.2 strain of the Omicron variant continues to spread, something I think we will probably hear about in today's presentation, overall, COVID-19 cases continue to decline globally. According to the latest data from the WHO, global cases fell by 21% in the week ending February 20th. This has led many countries to revisit their policies and restrictions on masking and social distancing, while still encouraging vaccination as the primary way to protect against all variants of concern identified so far. Just this week, European pharmaceutical companies, Sanofi and GSK, reported data on an updated version of their protein-based COVID-19 vaccine, which the companies say provides outstanding protection against severe disease and hospitalization, while also dramatically increasing antibody levels when used as a booster following other vaccines. The protein vaccine, originally developed by the companies with support from the US government's Operation Warp Speed program, provided disappointing results in clinical trials last year, but the companies are planning to seek authorization for this newer version in both the US and Europe. Before we get started with today's presentation, we'd like to once again congratulate the winners of the 2021 Michelson Prizes and invite you to join us for the virtual awards ceremony immediately following the next Global COVID Lab meeting on March 10th at 11 a.m. Eastern Time. The prize ceremony will feature Pulitzer Prize winner Laurie Garrett, a leading voice in the fight against COVID-19. Please go ahead and scan the QR code here to register for the event. It is my great pleasure to now introduce today's speaker, Dr. Salim Abdool Karim, Director of the Center for the AIDS Program of Research in South Africa, or CAPRISA, and Professor of Global Health at Columbia University. Dr. Karim is a clinical infectious diseases epidemiologist who is widely acknowledged for his scientific contributions to and leadership on AIDS and COVID-19. He is an adjunct professor at Cornell and Harvard universities and is pro-vice chancellor at the University of KwaZulu-Natal. He has served as president of the South African Medical Research Council and is a member of the WHO Science Council and the WHO TB HIV Task Force. He is also a member of the Africa Commission on COVID-19 and the Lancet Commission on COVID-19. During today's presentation, please send me your questions using the Q&A function in Zoom. Feel free to use the chat for general comments, but please don't post questions there. I will ask our speaker a broad selection of your questions after the presentation, and we will have about 20 minutes for discussion. With that, it is now my pleasure to introduce Dr. Karim.
Thank you very much, Kristen. It's a great pleasure and an honor to be with you here today. When I was asked to come and discuss Omicron and the epidemic in South Africa, I thought that this was particularly timely given where we are at the tail end, and we now have a clearer understanding of what we've been dealing with. So I would like to use this opportunity to give you a sort of quick update on the situation as it pertains to South Africa and the Omicron wave that we have just been through. So I'm going to talk about, to start with, why the concern about variants. Then I'm going to talk about the Omicron variant, particularly its 4 characteristics. And then I'll end off with COVID-19 in South Africa. What do we think is in store, or what might be in store up next? So when we think back that in the almost the whole first year of the pandemic, we really had just 2 variants, the original or ancestral variant, sometimes referred to from its source in Wuhan, and then that variant acquired one very important mutation at position 614. So the D614G variant then became dominant. It emerged in Europe and then spread throughout the world. So for much of 2020, we had just this one single variant, the D614G variant, spreading across the world. It was in December of 2020, on the 14th of December, that the UK announced that they had discovered the alpha variant. At that stage wasn't called the alpha variant, but essentially a variant with the 501Y mutation. 4 days later, in South Africa, we announced the discovery of the beta variant. And shortly thereafter, in January, we received the announcement from Brazil about the gamma variant, and then in March, the Delta variant, and then most recently, on the 25th of November, we had the announcement from South Africa about the Omicron variant, which had first been noted and loaded onto GISAID from Botswana, and hence the Botswana flag. Should be pointed out that the 5 variants of concern that we have had globally come from all 4 continents. So, I mean, 4 of the 6 continents. So we are seeing variants emerge all over the place. So why are we concerned about these variants? Well, let me just give you an example. Here I'm showing you the first waves in India, in Brazil, and in South Africa. In fact, in India, the first wave was so mild, politicians had said they had conquered this virus. But it didn't take long before what we saw was the emergence of variants. And the Delta variant caused a nasty wave in India. You may have seen the pictures on television, which were heart-wrenching, of people dying in the car parks because there's just no place in the hospitals. They were running short of oxygen. Everything was under huge strain. The gamma variant in Brazil led to a horrific second wave. And in South Africa, the beta variant led to our second wave, the Delta variant led to our third wave, and of course, most recently, Omicron to our 4th wave. So what we have seen is that variants are now driving the individual new waves that we are experiencing. And if you look at South Africa's situation, we had our first wave, which was driven by the D614G variant, subsequently our beta wave, gamma wave, and most recently Omicron. But what we have been seeing since then, in the recent past, is we have now started seeing a resurgence of cases. When we analyze where that resurgence of cases are, as we ended the Omicron wave, we now see that we are experiencing an increased number of cases in those who attend schools, because the schools opened in January. And with the opening of schools, we have now seen an uptick in cases in the adolescents, the 10 to 19-year-olds, and even in the younger ones as well. But what's interesting is, as you've already heard from Kristen, that while in December, the dominant version of Omicron in South Africa was BA1, in January, that has changed. We are now becoming dominantly BA2. Now, the 2 viruses are versions of Omicron. It's probably simplest to think of them that way. But when we look at the situation in South Africa, the red line reflects the proportion of all tests done on the platform, the Thermo Fisher platform that looks at the S gene. And so the red line reflects S gene target failures. And so what you're seeing is at the height of the Omicron wave, we were seeing a very high proportion of S gene target failures, reflecting BA1. Now, over the last few weeks, we have seen a steady decline to the point now where S gene target failures are less than 50% of the viruses that we are testing for, and using phylogenetics, we now know that those are all BA2. So BA2 is now, I think in the last week, comprises about 80% of all viral transmission in South Africa. Fortunately, when we look at this wave and what has happened over the last week, that the number of cases has now started coming back down. So the BA2 wave or resurgence that occurred when schools opened has now subsided, and we are back on our normal trajectory. And in South Africa, the epidemic is very clearly occurs in waves. In fact, the waves are so clear that it's quite predictable when we will get the next wave, because the interwave period is quite consistently about 3 months, ranges from 94 to 99 days. So it's pretty clear we can pretty much predict, and we did predict down to the day when the fourth wave would occur if there was a new variant. So given that we are now dealing with that situation, what is it we know about the Omicron variant and its genetic mutations? Well, I was first informed about the Omicron variant about 4 or 5 days before it was announced, when the first sequences were made available, and Professor Tulio de Oliveira's office, was one floor below mine, called me to come and have a look at this new variant that he's just identified. And when I looked at it, I mean, I was just taken aback, because it had mutations that were present in alpha, beta, gamma, and delta. So I looked at it and said, wow, this is like a super variant, because it draws on many of the mutations that confer important advantages to the previous 4 variants of concern. For example, it has the deletions at position 69 and 70 that were present in alpha, that led to alpha being an S gene target failure or S gene target negative. And that's the same mutation that gave Omicron the same characteristics. It has the K417N and even the mutations at 484 and 501 that confer a level of immune escape to the beta variant. So what we are seeing in Omicron is a whole range of variants, and the fact that it has over 50 mutations was quite striking. So the question is, so how did Omicron even come to exist? Well, we're not going to know that answer for quite a while, because we have no idea who patient 0 is. The initial outbreak, where the South African phylogeneticists identified Omicron as a new variant, was a small outbreak in university students in just outside Pretoria. But we don't know if they were patient 0. And in fact, even before we had identified those patients, as I pointed out, there was already a sequence of Omicron on the GISAID database from both Botswana and from Hong Kong. And the patient from Hong Kong was somebody who traveled from South Africa. So it was already present in other countries. And of course, subsequently, we now know that it was also present even before we knew it existed in South Africa or Southern Africa. It was already in existence in the Netherlands. So there are 3 broad hypotheses about how these variants come to exist, whether they occur, as a result of persistent infection in immunocompromised person, or the virus is circulating without detection, developing multiple mutations over time until it sort of surfaces with mutations that enable it to spread faster, or it's a transmission that occurred from animals, that the virus infected animals, and there are, of course, many animals that are infected with SARS-CoV-2, including domestic animals like cats and dogs. But when we look at the phylogenetic tree, we see that actually Omicron separates in the phylogenetic tree roughly in around September, October of 2020. So its ancestor was already in circulation in around September of 2020. And so it's quite likely, and this is just one of the possibilities, but it's quite likely that it infected an individual who was immunocompromised roughly in September 2020. And over the next 12, 13 months, it caused a persistent infection. And in the course of that persistent infection in an immunocompromised individual, we saw multiple mutations accumulate. And you would need to have a very long persistent infection to accumulate this number of mutations, because there are over 50 mutations in SARS-CoV-2. So that's one scenario. And so it probably emerged in around a year later, in around September, October of 2021. And by the time we first picked it up in South Africa in November, it had already been spreading for a few weeks. Well, is there any evidence to suggest any of these hypotheses? Well, there have been several studies now, and I've just provided you with this one where the Delta outbreak that we saw in Hong Kong was driven actually by hamsters. A sick hamster, infected hamster, infected a whole lot of other hamsters in this pet shop called Little Boss. And then the individuals who took those hamsters home got infected, and that's how we saw the spread of this virus in Hong Kong. So animal transmission is well documented. It's been documented in deers in New York State and so on. But here's a case of a highly, severely immunocompromised individual who is from South Africa. And this individual had HIV infection with a very low CD4 count. And this individual developed a persistent infection over 7 months. And this individual started off on day naught with the D614G variant that we were seeing, the standard variant that was in circulation at the time. But you can see how this individual accumulated critically important mutations. By day 6, this patient accumulated and acquired the mutation at position 484. By day 71, acquired the mutation at 417. And towards the end on day 190, acquired the 501 mutation. So 501, 484, and 417 are the 3 critically important mutations in the beta variant. So essentially what we're watching here is how a patient, through persistent infection, recreated the 3 key RBD mutations from the beta variant. And so that's one of the hypotheses about the likely scenario as to where Omicron might be coming from. And we've seen that patients who are immunocompromised for a whole range of reasons, whether it's leukemias or antiphospholipid syndrome or lymphomas, it's just a whole range. And so you have these immunocompromised individuals in countries across the globe. And so you can get these mutations and you can get these kinds of multiple mutations in the virus creating variants occurring anywhere across the globe. Now, it should be pointed out that because these individuals are severely immunocompromised, they aren't able to mount an immune response to the virus that other people would normally do. So for most people, they would just mount a response, clear the virus, like you and I. But because these individuals are immunocompromised, they aren't able to mount that immune response, so the virus becomes persistent. But that's the very reason why, even when you vaccinate them, they don't respond. And so they need multiple doses of vaccine before they can be protected from the virus. So let's deal now with transmissibility. How transmissible is the Omicron variant? So within the first week of the data that we had up to the 8th of December, we conveyed this in the Lancet that we, at that very early stage, the one thing that was becoming very clear was that Omicron is really very, very transmissible, much more so than any of the previous variants. So if you look at this is Gauteng province, one of the big provinces in South Africa, which has Johannesburg, you can see the first wave and the second wave, and then you can see the big Delta wave in yellow, and then you can see the red Omicron wave. And Omicron had a doubling time that was significantly faster than what we've seen with any of the previous three variants. And so we knew at that very early stage that we were dealing with a variant that was quite rapidly transmissible. Of course, we know now that it spreads much more rapidly, it spreads fast, so the number of cases accumulate fast. But because it spreads so fast, it consumes all the susceptibles quite rapidly. And so it comes down, the number of cases comes down fast and pretty much just as fast as it goes up. And so we've been seeing that across the globe, not just in South Africa. So why is that the case? Well, we now are seeing data emerging from multiple studies, including in animals, that the Omicron variant has changed somewhat from the previous variants. And one of the ways it's changed is that it has switched its entry route preference. So instead of going through the ACE2 tryptase and attaching, it can now enter cells through an endocytosis process. So this is a much more efficient way of entering cells. And so when it enters the cells of the nasal epithelium in this way, it can enter much more rapidly than the previous variants, which depended on the usual route of entry. But what we have seen particularly useful is that in cells of the nasal epithelium, the back of the throat, and the bronchus, that we are seeing a highly efficient entry process as a result. That Omicron is about 70 times more efficient in entering the cells of the bronchus and the nasal epithelium. But the price that the virus has paid in terms of fitness is that for alveolar cells in the lungs, that it's 10 times less efficient in entering these alveolar cells. And so when we look at the overall picture, we are seeing a virus that is very efficient in infecting cells of the nose, throat, and the bronchus. And so it spreads very rapidly. But because it doesn't infect the cells of the lung as efficiently, we've been seeing less respiratory distress. And so when we look at the symptom profile of Omicron compared to Delta, in Omicron, what we are seeing are many more cases of sore throat, fever, cough, really dominate the clinical spectrum of the patients we see in Omicron. And we are seeing far fewer cases, with systemic things and lung impact, like shortness of breath, irritated eyes, and loss of smell and loss of taste. In fact, in my own clinic, far fewer patients with Omicron complain about loss of smell and taste compared to what we saw with the beta variant, for example. So the clinical profile of what patients are presenting with has changed, driven by the biology, and based on its biology, its epidemiology has also changed in terms of its higher transmissibility. But this change in the clinical profile has had a huge impact. So here I'm presenting the last three waves in South Africa. So this is the beta wave, the Delta wave, and the Omicron wave. And this is the data from Gauteng province for the first 4 weeks in each of these waves. You can see how there were far more cases of Omicron in the first 4 weeks compared to beta and Delta. those waves were much more spread out. So it wasn't that Omicron necessarily caused many more cases, it just occurred more quickly. But what was interesting was if you look at the proportion of patients admitted to a hospital, it was significantly less at 5% compared to, for example, beta variant, where almost 1 out of 5 patients with beta were admitted to hospital. And we see that also in terms of oxygen requirement. We're seeing a much lower proportion of patients, almost half the requirement for oxygen. But I think probably the most significant measure is the proportion of cases that are severe. Severe being defined as patients in respiratory distress requiring oxygen or mechanical ventilation or intensive care admission or death. So we're seeing that with Omicron, we have a much smaller proportion of patients admitted who were severe. And that, of course, translated also to a much lower death rate. So overall, the clinical picture of what we're seeing with Omicron is a virus that is less severe in its clinical profile, causing fewer deaths. But because you get so many more admissions, the hospitals still come under pressure because the pressure on the hospitals is now, in a way, concertinaed into a smaller time frame. So let me deal now with immune escape with the Omicron variant. So one of the things that was noted was that the 2 key monoclonal antibodies used in treating COVID-19, that Omicron escapes neutralization by both these monoclonals. But it doesn't escape some of the other monoclonals, the more recent monoclonals, but certainly the 2 that are widely used were no longer effective against Omicron. So what about antibodies from past infection? Do they protect and do they neutralize the Omicron variant? So in this study published in the NEJM, these are individuals who acquired alpha variant infection. In the second diagram, these are individuals who had beta variant past infection. And in the third diagram, these are individuals who had Delta variant past infection. So if one looks at the convalescent serum from these individuals, and if you look at the IC50s, basically you are seeing very little ability to neutralize the Omicron variant, the B11529 variant, Omicron. And that's the same regardless of whether you previously had alpha, beta, or Delta. You had very little ability to neutralize the Omicron variant. And so the answer to this question is no. The past infection antibodies have little in the way of neutralization of the Omicron variant. And as a result, when Omicron came and hit us in South Africa, we saw a rapid rise in the number of reinfections. Now, we monitor reinfections. We have a process whereby an individual who tests positive on PCR, the system can identify whether the same individual was positive previously in the system. And if so, if they were infected positive more than 90 days before, we regard that as a reinfection. And so what we saw with Omicron is 2.4 times more reinfections occurring. So individuals who had past infection had little, if any, protection against Omicron. And that's part of the reason why Omicron spread so fast and so widely, because there's very little protection from past infection. But what about vaccination? Well, the team here did the study very early on. This is looking at neutralization of patients who received 2 doses of the Pfizer vaccine. And what was seen is an almost 40-fold reduction in neutralization against Omicron versus D614G. And the implications of that we are seeing is breakthrough infections occurring in South Africa's 4th wave. And again, in the same study in New England Journal of Medicine, they looked at the Moderna vaccine, the AstraZeneca vaccine, or the AstraZeneca vaccine combined with the Pfizer vaccine, or 2 doses of Pfizer. And across the board, the same situation. We see very little neutralization against the Omicron variant. And indeed, what we have been seeing across the board, a fair number of breakthrough infections. Now, in this analysis, which was done in the UK, because they are able to follow these patients up. So this is a longitudinal follow-up of individuals. And what they show, and let's just look now at the Delta variant, that efficacy against the Delta variant in individuals who'd received 2 doses of the Pfizer vaccine steadily deteriorated from around 95% initially, shortly after receiving the second dose, to close on to just over 60% when we look at the situation over time. And now what happens is when we give these individuals a booster dose, protection to Delta goes back up. In Omicron, it starts off lower, but it deteriorates to quite a low level. I mean, to very low levels of protection when you get to around 6 months after you've had your second dose of Pfizer vaccine. But when you get your booster dose, it boosts the antibody levels, and so it goes back up. And so we see neutralization occurring, and we're seeing better protection from clinical infection from Omicron when you've received a booster dose of the Pfizer vaccine. And you can, I will now share with you data that just brings together data from multiple sources. And I'm showing you here the AstraZeneca vaccine. So if you look at the AstraZeneca vaccine, that the original trial in the UK against the D614G variant, the AstraZeneca vaccine, 2 doses, provided 70% protection. The same vaccine tested in South Africa against the Beta variant showed very little protection, just around 10%. But studies in the UK showed that for the Delta variant, it did pretty well at around 60%. Against Omicron, very little protection. Less than 10%, almost no protection from having 2 doses of AstraZeneca. However, if you've had 2 doses of AstraZeneca and you take a Pfizer booster, your protection goes all the way back up to what you would have had if you just had your first 2 doses of AstraZeneca. So the boosters make a huge difference in protecting if you've had AstraZeneca vaccine before. Let's just compare that with the situation of the Pfizer-BioNTech vaccine. The original efficacy against the D614G variant in the US was 95%. The Pfizer vaccine was also tested in South Africa against the Beta variant and was also 95% protective in South Africa. However, in studies both in Canada and Israel, the Delta variant showed a reduction in vaccine efficacy of the Pfizer vaccine. Against Omicron, does really poorly at around 35%. But with a Pfizer booster, a 3rd dose, it goes all the way back up to 76%. So it just gives you some idea of how vaccine efficacy against clinical infection. So this is any clinical infection unrelated to severity, just any clinical disease. We see quite a big difference across variants and how variants are influencing the efficacy of our vaccines. And it differs based on which vaccines you're talking about. But the one thing that has been quite reassuring, and there are now several studies and data are now emerging on Omicron as well, what we are seeing is pretty good protection against severe disease and hospitalization. Actually, it doesn't matter which variant that you're looking at. In fact, it doesn't even matter how long ago you were vaccinated, that even up to a year after you received your second dose, you still have pretty good protection, usually in the 80s and 90% range, occasionally going down a bit, but pretty high levels of efficacy against severe disease and hospitalization. And so that's the one positive note that has come out of this, that we are seeing, regardless of variant, pretty good protection against severe disease. So let me end off with just making a final comment that when we look at the Omicron variant, it has driven the global fourth wave. So at a global level, here's the first wave, the second wave, the third wave, and now here's the Omicron fourth wave. It was in its own league. I mean, for much of January, the world was setting record numbers of new cases of infection because Omicron was spreading so rapidly and so widely across the universe. And so we are now recovering from this Omicron wave, and it's coming down just as fast as it went up. So if we look at South Africa, I'll just make some observations that might be helpful. So when we look at the peaks of the 4 waves, each wave has had a higher peak than the previous waves, and that's a reflection of higher transmissibility. So the beta variant was more transmissible than D614G, Delta variant was more transmissible than beta variant, and Omicron was more transmissible than Delta. And so what we have seen steadily is transmissibility has been important. But if you just take out the Omicron variant for now and just look at the trend in South Africa in our first 3 waves, well, not only were the peaks higher, but in each subsequent wave, we had more admissions, we had more cases, and we had more deaths. So the trend in the first 3 waves in South Africa was that the virus was getting worse. And we expected that the fourth would follow that trend, and that we are likely to see a fourth wave that would be even worse than our previous 3 waves. We were just fortunate, and I'm not sure whether it's just good fortune, good luck, or whatever it is, but that Omicron didn't have the same level of severity, even though it led to more infection, certainly among those who had past infection, and even in those who are vaccinated. We saw pretty high reinfection rates and breakthrough infection rates. So the question that begs itself, given that we can pretty much predict that within about 3 months, we're going to be seeing our next wave if the past trend continues. So what would that wave look like? And that's a difficult question because it's hard to predict. And the reason it's hard to predict is that each variant has been generated independently of the other waves. So they were all being generated almost in parallel with each other. So, the Delta wave didn't come from the beta wave. The Delta wave emerged and was probably being generated while the beta variant was being generated. And so what we are seeing now are variants that took longer to generate. So we don't know, because none of the variants follow the past variants. We have no idea what the next variant is going to look like. Is it going to be more like Omicron, or is it going to be more like Delta? Your guess is as good as mine. So it's very brave individuals who are predicting that, we are over the worst and the next variant is going to be very mild and so on. Vaccines do provide protection for severe disease, but they don't seem to provide much protection without boosters against Omicron. So it remains enigmatic as to what we can expect next. What we can expect, if past trends continue, we'll get our new wave towards the end of April, and that it's very likely to be driven by a new variant, because the Omicron variant is so widespread in its infection, and natural immunity to Omicron is protective against Omicron, and also protective actually against Delta. It stimulates Delta. It's almost a booster for those individuals who had Delta infection, and the antibodies against Omicron do pretty well at neutralizing Delta. So it's unlikely that the next wave will be Delta or Omicron because of the level of protection we have against those variants, but we have no idea what that new variant is going to look like. So watch that space. We'll have to figure it out when the new variant occurs. On that note, it's a great pleasure, and I'll stop there. Thank you very much.
Thank you so much, Dr. Karim, for that very clear and thorough presentation. Very much appreciate it. And we have many questions coming in for you, so maybe we'll jump right in and start with those. So one of the first questions is, what do we know about how the severity or the reduced severity of Omicron is attributable to acquired immunity versus the actual biology of the variant?
Right. So I've been asked that question on multiple occasions as to, what is driving the lower severity? Is it that the virus itself biologically is less virulent? Is it that there's past immunity from natural infection? Or is it that people are vaccinated? So I think it's quite likely that all three contribute.
Mm-hmm.
And depending on vaccination levels, they vary in different settings. In South Africa, it's probably because vaccination coverage is still at around 40% of adults. And when we were hit by the Omicron wave, we were around 30% of adults. But we've had pretty decent vaccine coverage of the elderly. And the elderly, we are over 60, 70%. So vaccination would have contributed. The lower virulence of the virus probably contributed. And we've had a wave after wave of beta and D614G and Delta. So it's probably some level of past immunity has also been contributing, especially to a situation of preventing severe cases and death.
Right, right. So we have a question on the ongoing zoonotic transmissions, and with the hamster event in Hong Kong and the knowledge that the sewage in New York City is replete with persistent non-human variants of COVID-19. How do we manage the pandemic with this ongoing transmission? And is there any serious strategy for this?
So it's almost a no-win situation, unfortunately. But I'm often asked, so what do we do next? And there's no right answer to that question. But the one thing that does occur to me, that is that even though the coronavirus has a pretty long genome and that it can generate many new variants, there has to be a point where it cannot continue to mutate to advantage, that the mutations don't really confer functional advantage to the virus. And so that would mean that those new mutations that continue to occur, the new variants that continue to occur, they just don't confer sufficient advantage. And so when we get to that variant, that probably is like the super variant. It can't really get any faster than that variant. It doesn't have any more advantages than that variant. Then that variant is probably going to be the one we stuck with. So that's sometimes referred to as, that's the endemic variant that we will have to learn to live with. And when we reach that point, we'll be in a much better position to make a better generation of vaccines, because now we know what the variant is, like measles, we know what it is. It's not going to go and change itself into some new variant. And so at that point, we will be in a much better position to take hold of that situation, keep it under control. And even if it gets jumps from animals, it would be very difficult to beat that dominant endemic variant. And so as long as we're in that situation, we will pretty much be able to control it. Right. Now, some have said that endemic variant is going to be a very mild variant. Mm-hmm. Well, there's no basis for that, except that the other 4 coronavirus variants that have reached endemicity are all very mild. In fact, we don't even bother about them because they just cause a mild cold. So if SARS-CoV-2 follows that same trajectory, yes, it could be mild, but in my view, it could just as easily not be mild. We just have no way of telling. So how do you manage the situation going forward? I think it's really about 3 things. First, you've got to have surveillance. Mm-hmm. You can't deal with a variant if you don't know it's there. You've got to have surveillance. So in South Africa, we have a pretty good surveillance system. That's why we've identified 2 of the 5 variants. Right. Not necessarily that they emerge there, but that's because we have the systems in place for it. So you've got to have the surveillance, and the sewer surveillance, sewer pads is really important as well. The second is that you've got to adjust your prevention measures in relation to the variant. So when you don't know what the variant's going to do, you've got to take some precautions. So you've got to go vaccine plus combination prevention, institute masks or whatever you need in terms of public health measures as a precaution until you know much more clearly what this variant is doing. And so that gets me to the third issue, that we need to be able to quickly assess when a new variant emerges. Do our vaccines protect against it? Does natural immunity protect against it? We need to have a panel of these blood specimens and ready to go so that we can get an answer quickly. So it takes out the uncertainty. And we're very clear about what our strategy can be. And I think we're going to get better and better at that. So we'll manage the next variants better, and we'll have a shorter period of uncertainty.
Right. Okay. Thank you so much for that. So we have a question on how the youthful skewing of demographics in South Africa affect the epidemiology and the clinical outcomes for BA1 and BA2. And is it necessarily applicable to older societies in Europe and North America?
So the initial wave, as Omicron started spreading, the original BA1, was also, we saw a disproportionately higher proportion of tests in the very little ones, below 10 and even up to 20. And we also saw more hospitalizations in the younger ones. Now, I'm not sure why that was the case, but 2 things became apparent. The first is that even though we saw more hospital admissions of the children, they were really very mild. They weren't severe cases. We had very few severe cases. So even though we had more hospital admissions, they were not more severe. The second is that, some people tell me, and I don't know if it's true, that because infection in the upper respiratory tract in children is in a smaller space, they get the congestion and the symptoms much more readily than the older kids. So upper respiratory tract infections impact children and babies much more than they do in adults. The adults just deal with it. So I'm not sure if that's the case, but anecdotally, that's what I'm told. So whatever the reason is, but it's true, we have seen it, and it's occurred in Europe as well. So it's not an African phenomenon, no.
Sure. And how is that in people living with HIV who are on long-term antiretroviral therapy? How are they responding to the older people living with HIV? How are they responding to Omicron infection? And what is an appropriate vaccine regimen for older people living with HIV who are on antiretroviral therapy?
So we have generally been seeing, and we've now done about 3 clinical trials of vaccination in HIV positive individuals who are obviously on antiretroviral therapy. So most, they all are immunosuppressed. They were all virally suppressed, not immunosuppressed, they're virally suppressed. So they do pretty well. We see a pretty good antibody response comparable to HIV negative individuals after the second dose. It's not as good against the first dose, but after the second dose, they do pretty well. So we've had no concerns about, because we have close on to 8 million people in South Africa who have HIV infection. So obviously it bothers us. We have to establish that. And so we've been working very much on the basis that people on ART are responding very much like people who are negative, and they do just as well with 2 doses. And they've been given 3 doses, just like everyone else, if they've been 4 months since their second dose of Pfizer.
Okay. So what is the right number of doses for those who are immunocompromised? Is it 3? Is it 4? What is the...
Oh, the answer to that is actually easy.
Okay.
As many as it takes. You've got to just keep giving them doses.
Right.
But the thing is, you have to monitor whether they're actually responding. It turns out that in transplant recipients, that 3rd and 4th doses seem to do the most.
Okay.
So you're probably going to need about 4 doses if transplant recipients are anything to go by. But we've not done those studies, and I don't know if anybody has, as to how many more we're going to need. And I guess it's probably going to be more personalized, individualized. Patient X will respond differently to patient Y. So you've got to vaccinate, look for the immune response in terms of antibody levels. If it's not there, give them another dose. It's not there, just keep giving them doses until they respond.
Okay. So 2 other vaccine-related questions, and one is on what you think about developing Omicron-specific booster vaccines, and also the approach of intranasal vaccines as potential boosters in the future.
Both are excellent questions. I was quite taken aback when I saw the preprint that came out last week from the VRC, where they took the Moderna vaccine and made a Moderna vaccine of the Omicron sequence. And they challenged non-human primates. They vaccinated them. So these are non-human primates that had been given the Moderna vaccine previously. So they were now boosted either with the original Moderna vaccine, or they were boosted with the Omicron version of the Moderna vaccine. Made no difference. In fact, the 2 lines, the original vaccine does a little bit better. I mean, not meaningfully better, but basically makes no difference. So it doesn't seem that there's any value, certainly from a non-human primate study. It might be different in humans, but from a non-human primate study, that doesn't look like having an Omicron-specific vaccine makes much difference. Also, I think the way it seems that the level, the titer of antibody is more important than the actual nature of the antibody. Well, the avidity is important as well, but the level of the antibodies become important, and that's why boosters become important. And in fact, in South Africa, we are in a situation, because we got vaccines late, because of global inequity in vaccine distribution, we have quite a high proportion of our population had already been infected. So many of the people who are vaccinated in South Africa actually don't have vaccine immunity, they have hybrid immunity. They have both natural immunity and vaccine immunity, which is the best kind to have, actually. So not that I'm promoting getting infected, but for those who did become infected, it's pretty good. So I hope that helps in answering the question, that actually it doesn't look like variant-specific vaccines are going to be useful. As far as intranasal, I was impressed with the Yale study. I'm assuming that that's what they're referring to. I mean, that intranasal vaccine, the responses actually were able to neutralize quite a range of the Sabeco viruses. So that's pretty good. But, let's wait to see. I'd say watch this space rather than make any commitment to it.
Right. So we have a question specifically on the Omicron surge in Hong Kong. And has that been characterized as a BA1 or BA2? Do we know?
That's a good question. I haven't checked. So I don't know. I don't know the answer to that.
Okay.
But I mean, the situation is horrific in Hong Kong. Wow. I mean, 7,500 cases. I mean, for a country, we call it a country, yeah. For a country that hasn't had even 1,000 infections ever until 2 weeks ago, to have 7,500 cases a day. I mean, the hospitals are just, yeah. So looking at it, it's quite likely to be a BA2. Well, clinically, it doesn't look like they differ that much. I know there is a study that suggests that BA2 might be more severe, but it's questionable. The data we have in a large data set suggests no real difference between BA2 and BA1. So I'm not expecting it to be particularly different. Then and in South Africa, we've treated it very much as BA2 is not another variant. It's just more Omicron, just faster spreading Omicron.
Right, just what we need. So we had a question on if you could speculate why Omicron-specific immunity wanes at a greater rate after 2 doses of the Pfizer vaccine than Delta-specific immunity, which I'm guessing has to do with the antibody escape mutations, but I'm going to let you answer.
I think it relates to the drop in neutralization. So with Omicron, the drop is drastic, at 40-fold or so, if somebody had 2 doses of Pfizer. With Delta, it's less. So in other words, the titer of antibodies you need to effectively neutralize Delta is a lower threshold than for Omicron. You need a higher level. So that's why those boosters become so important. Because once you boost, you're getting about 75-fold increase in your antibody titers. So I would guess that that's the main reason in all likelihood.
Right. So we have one question on some of the data that's coming out of Israel with the 4th booster and showing that maybe it doesn't provide any greater immunity or protection necessarily. So are we reaching the end of the line with what we can do with boosters of the same vaccine?
Yeah, I have to say I'm not sure about those data just yet. I think it's early, so I'm not going that route. But you can't just keep vaccinating. It's a point where you just keep vaccinating. You just get a boost. You'll get a small boost. It'll be temporary. It'll dissipate. The 3rd dose seems to make quite a big difference, and I would expect that benefit will taper quite drastically thereafter. And that's one of the reasons why most of our vaccines, I mean, if you take, for example, DPT, we give 3 doses. Doesn't make any much point in giving a 4th dose. I mean, they are hepatitis B, we give 3 doses. So I think I'm not expecting that 4th, 5th, 6th doses are going to make much difference. But I think that one of let me just make a point. It's actually not really any public health intervention to go around giving doses every few months, to keep giving people doses. That's not how you're going to deal with this problem, because it's not really practical. It can't be done on any grand scale. Might be able to do it in Israel because they have access to vaccines while the rest of us don't. But it's very hard to do it beyond that. So I'm not anticipating that we will see this. I think what we're going to need to do is we're going to need to move to the next generation of vaccines. And that next generation, I'm anticipating, would be more vaccines that are pan-coronavirus vaccines, vaccines that focus on the conserved epitopes. So that's what I anticipate.
Yeah. Well, I think that's a perfect place to close with the hope for the future. And I just want to thank you so much, Dr. Karim, for today's clear and engaging presentation. We really appreciate your taking the time to share this work with our audience and for answering so many questions. So thank you so much. I would also like to thank all of our attendees for participating today and for submitting such great questions for our speaker. With that, I would invite you to join us 2 weeks from today on March 10th. Our speaker that day will be Dr. Chris Chu, a professor of infectious diseases at Imperial College London, and he will present on SARS-CoV-2 human challenge studies, a novel strategy to accelerate vaccine development. And immediately following the lab meeting will be the virtual awards ceremony for the 2021 Michelson Prize winners. So please plan on joining us for both events that day. And if you're interested in more research on COVID-19, please sign up for the HVP COVID Report, a bi-weekly newsletter that provides insights from experts around the globe and highlights the latest scientific articles and data. With that, I'll say thank you again for participating today. Stay safe, and we hope that you'll join us again for the next Global COVID Lab meeting. Thank you so much.